Welcome everyone to this edition of MS Imaging Special Interest Group, brought to you by at the Albuquerque Airport. So apologies if you hear announcements. Today we have a great set of speakers who will focus on the analysis of small regions of interest. I'm Colleen Crouch, an assistant professor in biomedical engineering at the University of Tennessee, Knoxville. I am joined by my co-chair, Dr. Jessica Moore, who will be facilitating the Q&A since I have to leave a bit early. Jessica, can you introduce yourself?
Yeah, thanks everybody for joining us. My name is Jessica Moore, and I'm a senior scientist in proteomics at Discovery Life Sciences, and I'm here to help facilitate today's session.
And today's topic is focused on measuring small foci. An ongoing challenge in improving spatially resolved omics is how to measure small regions of interest. So today's session highlights two different techniques to accomplish this goal. Our first speaker is using stem cell technologies microscopy-based immunocapture system to select subcellular regions of interest under a microscope. And our second speaker's work focuses on tissue expansion, a subfield of expansion microscopy. This technique physically makes the tissue larger to enable high spatial resolution imaging mass spectrometry.
So for our agenda today, we'll have two presentations from our speakers, and then it will be followed by Q&A for each speaker, and then a fireside chat with the presenters, which will include more opportunity for discussion and questions if we have time.
So I'm very excited to introduce our first speaker, Dr. Christiana Lungu, who is a senior postdoc in the Department of Molecular Tumor Cell Biology at the University of Stuttgart, where she has worked since 2017. She earned her PhD in molecular epigenetics from the University of Stuttgart, and since 2023, she leads the spatial epigenetics research focus area within the department. Her research focuses on the spatio-temporal regulation of cell signaling networks, with particular emphasis on epigenetic enzymes, their interplay with signaling pathways, and their dysregulation in cancer contexts. Take it away, Dr. Lungu.
Thank you so much. Just share my slides. Right. Maybe again. Now it should work, right?
Yes, looks great.
Yes. All right. Yeah, so thank you so much for the invitation. It's really wonderful to share our research in this community. So I will be talking about spatial proteomics and why this is important for us to better understand how cells are organized and they work, in particular in the context of cancer. So spatial context is essential for cellular function, as I'm sure many of you know. And this is something that becomes immediately obvious when looking at the structural organization of our healthy tissues, which is not random. Now, this theme of organization is continued at the subcellular level in the form of organelles, such as the nuclei, for example. And these are membrane-bound compartments that are used to separate pathways that perform a certain cellular function. Now, in the last couple of years, there is a new type of cellular arrangement that has drawn a lot of attention. These are commonly called biomolecular condensates, and the nucleoli are a prominent example of such structures. Now, these arrangements are thought to be very important for the way in which the cells work. However, because they lack a classical membrane and they are usually very small, it is very difficult to study this by classical methodologies. So to be able to understand how cellular structure is relating to cellular function, one needs methods that allow us to access exactly these two types of information together. And this is where spatial proteomics comes into place. So for the next couple of minutes, I would like to share with you how we use a new spatial proteomics technology, which is called optoproteomics, in the context of breast cancer cells, specifically to unbiasedly resolve the proteome of small nuclear bodies that we have observed in these breast cancer cells. So before I go into the data, I would just like to give a brief motivation of what drives our work. So we are interested in how epigenetic enzymes are deregulated in cancer. And particularly interesting now for this talk is the so-called polycomb repressive complex 2. Now, this is a master regulator of cellular differentiation that, as its name already shows, is silencing gene expression. And the complex contains several subunits, so-called core subunits, out of which EZH2 is the catalytic subunit. And what this does is to trimethylate histone 3 at lysine 27. This plays an essential role in development. So EZH2 or PRC2 knockout, this is embryonically lethal and plays an important role, for example, in X chromosome inactivation. Now, frequently, as it frequently happens, this type of complexes are also exploited to drive disease. And now in the context of our work, we are focusing on triple negative breast cancer. So this is a breast cancer subtype that is actually the most aggressive and also the most difficult to treat. So what we know from before is that EZH2 is upregulated in breast cancer in general, but in particularly also in TNBC. And high levels of EZH2 are associated with a bad prognosis. So this has driven a very strong interest in the development of EZH2 and PRC2 inhibitors for clinical therapy. However, it became recently clear that inhibition of polycomb or EZH2 has a plethora of effects ranging from tumor suppressive to oncogenic. So this means that even if we are studying the biochemistry of this complex now for many, many years, we still do not understand how the polycomb complex is regulated in the context of the living cells, in particularly now in a cancer context. So the hypothesis that is driving this project is that the cellular context regulates the function of PRC2 in breast cancer cells. So to address this hypothesis, we focused on model systems that allowed us to address two things. First of all, we wanted to have model systems that are physiologically relevant. And for this, we use a system that recapitulates the metastasis of triple negative breast cancer cells from the breast to the bone, because it is known that PRC2 plays an important role in this process. And the other criteria was that these cell systems have to be compatible with easy manipulation to be able to study this in vitro. And now in TNBC research, the MDA NB231 cell line is frequently used. And the nice thing with this cell line is that this has been previously injected into mice, and the subpopulation of cells that form metastasis in the bone was isolated. So now this subline, this is genetically identical to the parental population, but has a higher tendency to metastasize to the bone. So it represents a different physiological context. And because we wanted to understand how space regulates PRC2 localization and function, the first thing that we did was to investigate the subcellular distribution of the complex in the cells by using high resolution microscopy. So we look here at the endogenous localization of SUS12. This is one of the core subunits of PRC2 in the parental cells. And what we see here is that the protein is localized in the nuclei of the cells, as expected for a classical epigenetic regulator. However, we were very surprised to see that for the bone metastatic subclone, so this cell line here, we have a subset of PRC2 that accumulates in these nuclear bodies. These bodies were around 1 micrometer in diameter and were observed in around 40% of the cell population. Furthermore, they also overlapped with the catalytic marks, so H3K27 trimethyl, indicating that these are not aggregates, but they actually play a role in the cell. So then the next thing was the hypothesis that if we understand what is the context, the proteomic content of these clusters, we can also understand what drives this context-specific function of PRC2. And this is now where optoproteomics came into play. So this is a technology that was developed by the company CinCell. And we have applied this now for the first time together to study the local proteome of epigenetic regulators. So in brief, the technology has 5 key steps. The first step involves the fixation of the samples as for a classical immunofluorescence experiment. After this, the sample is incubated with a photoactivatable biotin. And then a 2-photon laser is used to induce local bioaccumulation in regions of interest. And now because of the 2-photon laser, also the spatial resolution is extremely high. This is done, of course, in many cycles to be able to label enough material. So this is done also automatically on a microscope, after which the cells are lysed and then a biotin IP is performed, and then the samples are sent for mass spectrometry to a different partner. So we have done this in the context of PRC2 now by using EZH2, the catalytic subunit, as a marker. And here I would like to highlight that this is now the endogenous EZH2. So we do not perturb the native state of a system. And what you see here is that in these two cells, EZH2 forms this foci. And this is also what we use to define as a region of interest. And then here you have an example of the photobioaccumulation reaction, here from the top view and here from the side view. So what you see in red, this is the total EZH2 in this particular cell. And what you see in green is actually the subpopulation that was labeled and that is used later for mass spectrometry. So you can also, I think, here see reasonably well that we really look at very small structures, and this also allows us to fish out reactions that are very specific locally. So we have done this labeling in 4 biological repeats, and we have obtained an overlap of around 50% in the targets. And then we have further ranked them based on their enrichment and the quality of the peptides. And what you see here plotted, these are all hits that were detected with good quality in all of the 4 biological repeats. And it was nice to see that we can detect the core subunit of the EZH2 or the polycomb complex, indicating that the method gives rise to a result that makes sense. And then if we now look at the most enriched targets, we stumbled across PHF19 as being particularly enriched in the label sample. We also validated this, so we looked at the endogenous distribution of PHF19 in the 2 cell lines. This also forms clusters in the bone metastatic subline. And also these clusters are colocalizing with the PRC2 core subunits. So then we knew that this is something that is worth following up. So now what is PHF19? Now I was saying that PRC2 is responsible for silencing gene expression by trimethylating histone 3 on lysine 27. And however, the story is much more complicated than that. And this is something that we are only starting to understand in the last years. So the polycomb complex can be divided into more subtypes, specifically PRC 2.1 and 2.2. And the identity of the subtypes is determined by so-called accessory subunits. Now, PHF19 is one such subunit. The difference with these subunits is that they are substoichiometric. So this means that only a subpopulation of the polycomb complexes within one cell will contain these subunits. And these factors are there to locally tune where the polycomb complex will go. So you can think about the core subunit as the motor of a car and as the accessory subunits being the wheels that decide where the car is going. What was also interesting for us is that PHF19 has not been studied in the context of triple-negative breast cancer so far. So this is why we further continued to look at this protein. So the quick question was, of course, to try to understand whether PHF19 is important for the structure of these bodies. So we have knocked it down, and we have observed that the PRC2 bodies completely disappear. And this was not because the subunits of the polycomb complex were affected in their expression. The next thing was also to see whether these observations are conserved in other systems. So we also checked the TCGA database and looked at different breast cancer subtype patients for the expression of PHF19. And we have observed that particularly in the basal-like subtype, so this is now also called TNBC, this is where PHF19 is highly expressed. So we screened for more cell lines that recapitulate the subtype, and we found a second one that has this really beautiful polycomb foci. Then the hypothesis was that perhaps we can discriminate in between cells that have the foci versus cells that do not show the foci by PHF19 expression level, and this is indeed the case. So we see that the expression level of PHF19 plays an active role there too. So then we also use the second cell line to deplete PHF19 and check whether the polycomb complex foci still are present or not. And you can also appreciate here that the complex, that the foci completely dissolve. And again, this has nothing to do with the integrity of the complex itself. Then turning it the other way around, we have also used cell lines where PHF19 endogenous levels are low and the polycomb complex is diffusely localized. And then we have transfected back PHF19, which by itself forms this beautiful foci. And also we can see that it can de novo induce the formation of polycomb bodies at the exact same position. So we know that this is now also sufficient to drive polycomb clustering, polycomb organization in cells. So now the next thing, now beyond the general interest into how these clusters are formed and so on, we were also interested to figure out what is actually the role in cells, in particularly now in our triple negative breast cancer cells. So what we did there, then we have used these three cell lines. So as a reminder, these are the two that have polycomb bodies, and this is the parental cell line that doesn't have polycomb bodies. And we have depleted PHF19 here. And we checked for the motility of the cells because polycomb expression has been associated with invasive cell motility in TNBC. And what we have seen is that if we deplete PHF19 in the parental cell line, not much is happening. Although PHF19 is expressed there, however, polycomb is not clustered. Now, however, if we do this in the context of the cells where polycomb forms foci, cell motility is depleted. Now, keep in mind, we only perturb the accessory subunit. We do not engage here with the rest of the complex. So it's only about the local organization of polycomb into these clusters. Of course, such results can also be caused by the expression of PHF19, not necessarily also by the role of PHF19 in this clustering phenotype. So then what we have done next was to bring back PHF19 in cells that have low levels of expression. And we have done this either by overexpressing the wild-type protein or by using a mutant that lacks a domain which we identified to be important for this clustering. And then what we see here is that only when PHF19 is able to form foci and drive polycomb body formation, cell motility is promoted, and not when the protein is expressed but localized diffusely. So with this, I would like to, I'm already coming to the end of the presentation. I would like to share with you the model that we are currently working on. So what we have found to a large extent with the help of optoproteomics is that in triple negative breast cancer cells, PHF19, which is a polycomb accessory subunit, is highly expressed. And this drives the reorganization of polycomb PRC2 in the cell nuclei. We also, meanwhile, know that to do this, PHF19 engages several of its domains. So we know that the DNA binding domain is important. We know that the intrinsically disordered region is also important. And this organization by itself plays an active role in driving cell motility, which of course is particularly important in the context of cancer. Now, all of this would not have been possible to investigate without optoproteomics, taking into account that PHF19 has not been described so far in the context of TNBC. So with this, I would like to sum up and thank everybody who was involved in this work, the head of our department, of course, for all the support, very talented students that have generated a lot of the data, the collaborators from CinCell and our sequencing partners, and of course, our funders. And thank you very much for your attention. I think we have 5 minutes for questions, as far as I know.
Yes, that's perfect. So thank you for that amazing talk. I think it really did a great job of highlighting the power of this workflow and optoproteomics for, this type of analysis. So we do have time for audience questions. And if you do not mind, please use the Q&A box that's at the bottom of your screen to submit any questions, and we'll start to ask them as they come in. But as we wait, I will go ahead and maybe give you a couple of questions. And one of them is for this particular audience, US HUPO, right? A lot of these people are doing proteomics. And so for this particular technology, how small can you go? Like you're going subcellular, and then have you found any drawbacks or anything like that whenever you use these workflows?
Yes, so I think this is, I think now from the perspective of CinCell, one of the reasons why they were so interested in doing this together, as far as I know, I think these were at the time the smallest structures that they were testing in terms of labeling. So we had 1 micrometer size foci. And we did this in the first cell line when we have identified this foci. Now you have to calculate around 40% of the cells had these bodies, and we had around 2 to 3 bodies per cell. So there was a lot of instrument work time that went in this. From what I know, I think the microscope was running 4 days, like 24 hours to get enough material. So I think this is one of the things to keep in mind. This is also why the experiment has done on 4 biological repeats, because these are low abundance targets, and we wanted to make sure that we only focus on really the most enriched and specific targets. And I can also imagine that from the chemistry itself, the smaller the structure, because the biotin can also locally diffuse. So this can also be a challenge, but I think this is also where they have improved meanwhile quite a bit. So I think now in terms of size, I would just, I think maybe 0.5 micrometers is where they can do now a really good job. And after that, I think it gets really complicated.
Yeah, absolutely. And this community is mostly an imaging community, and we usually use like multi-mass spectrometry, which historically does a very phenomenal job, and it has some drawbacks, right? That we get like a single laser shot, and then those are the peptides or the proteins that you measure. But you have the ability to leverage the power of LCMS.
Yes.
And so is that a topic that you could speak to in terms of like the sensitivity you get? And especially, we're coming off of ASMS, where we just saw a host of instrumentation advancements in terms of sensitivity, speed, and low input from both Brooker and Thermo, and how you can maybe see this technology leveraging those new instruments that we're also very excited about as we come back to our laboratories.
So as far as I understood, I mean, I do not use any of this directly. So we have done the labeling in collaboration. What I regularly do in the lab is really imaging, as in microscopy. But I think one of the advantages now of this optoproteomics approach is that because you use a 2-photon laser, the labeling in the Z direction is very precise. So you do not have a column like you have for microdissection. Also, the laser is very thin. So then in the end, you really spend much more time focusing on the real targets and have much less background. Yeah.
And then maybe my final question would be, you already had a great antibody, and so maybe you could discuss like that with this technology, and maybe if you didn't, what you might do.
Yes, yeah. So indeed, we were lucky to have a really good antibody. However, also there, the staining had to be optimized just to make sure that there is not so much background labeling, and the contrast and so on is optimum. Now that the methodology works with anything that localizes to a specific region. So this means you can have a GFP tagged protein. This would work equally well. You can have this being either overexpressed or being a CRISPR knock-in. Yeah, this would be everything that you can see, no matter how you see it, either to staining or through genetic engineering, this would work.
Awesome. Well, thank you for that, and for the amazing first half of the talk.
Thank you.
And yeah. And with that, for the second half of the webinar, we're going to change gears a bit and switch to a graduate student from the laboratory of Dr. Boone Prentice, and that's from the Department of Chemistry at the University of Florida. Jacob completed his undergrad degree in neuroscience at Vanderbilt University in 2016. And then he went on to the Boston University School of Medicine to complete a master's degree in biomedical forensic science in 2018. And after that, he spent 4 years as a postmortem toxicologist with the Miami-Dade Medical Examiner. So super impressive background. And he joined the Prentice Group in the fall of 2022. And today he's going to share some really interesting information about how if you have a small foci, maybe you make it bigger to be able to measure it. So with that, I'll hand it on over to Jacob.
Okay, bring in my slides. And thank you for that introduction. Does everything look okay?
Yep.
Okay, yep.
You're not quite in presenter mode. Oh, there it is. Got it.
Yep, yeah, there we go. Okay. But yeah, thank you so much for that introduction, and thank you for everyone here, hear me talk about my research. As you said, I'm in the Practice Lab. I'm going to talk a little bit about expansion imaging mass spectrometry. Yep, there we go. Start off just like a really brief introduction on IMS, just in case people are not as familiar. Why you'd use it potentially versus other imaging modalities. On the left, we have maybe things people may be more familiar with, although it sounds like everyone's maybe more familiar with IMS. But I would posit its main strength, of course, is molecular specificity. We heard a lot about microscopy and fluorescence, and these are, of course, very powerful techniques in which you can stain and label a variety of protein structures, etc. But IMS, I think we all know, goes one step further in the ability to analyze and detect a wide variety of compounds with small chemical changes in them, simultaneously, which is, I think is, the poster child of this is kind of lipidomics, right? So we can show this is PC3040, phosphatidylcholine, and there's not really a great way to stain or label specifically phosphatidylcholine. You can do membrane stains, you can metabolically label potentially the class of PCs, but specifically PC3040 we can image, and we have an IMS image here, and this could be our red color here. But like I said, small chemical change, so PC3041, change of 2 Dalton. So any mass analyzer could resolve this too. This could be our blue color, add 2 carbons, PC361. Again, not really an issue for imaging mass spec. Some other strengths, of course, label free, really important for lipidomics, proteomics, etc. Any of these peaks can become an image potentially. And of course, as I was talking about PCs, if you're not familiar with lipidomics, there's of course a whole variety of PCs, not to mention any lipid class like single myelins, PEs, sulfatides, etc. So our ability to see multiple classes and multiple variations within those classes at the same time, of course, is a major strength. As well, of course, we know that can be used for a wide range of analytes, has been shown, throughout the literature, throughout the years. But of course, I want to focus on spatial resolution. And unfortunately, compared to maybe some other imaging modalities, especially microscopy, IMS is relatively poor in spatial resolution. Reading papers or seeing presentations, routine IMS will be anywhere from 30 to 70 micron, but I think maybe more recently, we're seeing a lot more at 5 to 15 micron, which really represents the commercial limits of several popular instruments, like 5 micron is the timsTOF, for example. In our lab, we have a 7 Tesla Solarix ICR using the SmartBeam 2 laser. That's a 15 micron limit on how our resolution. And so just to kind of demonstrate that, I have a transverse mouse grain section here. I'm highlighting PC361 at M/Z788, and just 3 images kind of showing what 80 micron to 15 micron looks like on our ICR. And you can see we're very tissue structure regions, right? So this is the cerebellum, and we have these distinct cell layers, white matter, granular, molecular. 80 microns, very pixelated, but getting down to 15, you can see a lot more detail here. But this kind of puts our high spatial resolution, or people who are trying to push this, really below 5 micron. In some biological context, the average mammalian cell is 10 micron, and we can actually see a single cell layer here at our 15 micron, but this is the Purkinje cell layer. It's a very big cell layer as far as it goes. So really, for routine work with our commercial instrumentation, we're kind of set outside of single cell analysis, although it is getting more popular and there are more papers trying to push there. And and more importantly, I forgot. Yeah, and again, compared to microscopy, this is an H&E stain. This is on our Zeiss microscope, poor spatial resolution. This is 5x objective, so this is 1 to 2 micron. We can, I think, appreciate the detail we can see in this versus our 15 micron. But as I was saying, IMS is kind of on the precipice of single cell imaging, and if we can kind of push our resolution even further, single cell can hopefully be routinely done, as well as potentially some smaller organelles, larger organelles, such as nucleus, mitochondria, things, in the micron range. So this is kind of what we hope to potentially achieve by pushing our resolution. So you might ask, well, what limits our resolution? So how we typically do IMS in apprentice lab is, which I assume is pretty typical. And as you mentioned, MALDI IMS is what we do. So we section with a cryostat. This will be on thaw mounted onto microscope slides. We apply our matrix with robotic sprayer or sublimation. It's more suitable for high spatial resolution imaging, and then we perform MALDI as such, and we can raster across the tissue, generating a spectrum at each point, and we'll get a point. And if we want to create an IMS image, any of these peaks and create a heat map of intensity, recapitulating the form of the brain. But the key thing we want to focus on is, of course, then the laser ablation mark size, right? So this really comes down to focusing our laser beam. That really determines our spatial resolution here. But there are some other factors. We'll see the research largely focused on the laser beam, but we also need to consider delocalization during sample preparation. As I mentioned, it's mostly just matrix application, but there is also other sample preparation you can do, sample handling steps. And during that time, during temperature changes, etc., maybe exposure to solvent, you don't want whatever you're looking at to not reflect the endogenous or native distribution of that analyte. My diagram previously was not completely accurate. I showed the laser hopping around, but in reality, we know the stage moves around, which means that you're reporting to say, oh, this is a 3-micron spatial resolution image or pixel size raster width. Not only does your laser ablation mark need to be smaller than that 3-micron, your stage needs to accurately be able to move 3-micron every single time. Lastly, and this is true of any type of high spatial resolution application, there's sensitivity is always a concern since you're sampling smaller and smaller amounts of material. And in the context of here, again, MALDI IMS, not only is abundance a concern, but also does it ionize well with MALDI, with our laser. So some research that kind of goes into pushing the resolution. This is just a sampling, and I think it's beyond the scope of the talk to go specifically into what these are doing. But like I said, they focus on either providing different geometries for the laser or using different wavelengths, adding fiber optics, or an additional laser for sensitivity, which is really to make the larger point that these are kind of expensive instrument grade modifications. Specifically, this is what T-MALDI2 looks like, transmission mode MALDI2. So if you're familiar, of course, like metal box or vacuum chamber would be more of the mass spec you purchased commercial portion, but then of course the transmission geometry would have to be itself, as well as implementing the Salkin laser, MALDI2 along with those optics. So while some of these are becoming maybe a little more commercially available, like MALDI2 is available on some Bruker instruments now, and SMALDI you can buy a SMALDI source. For the most part, these types of modifications are really not accessible to typical IMS lab. I kept using the word like routine analysis. What can most labs do, unless you're focused on instrumentation or these can be exorbitant in cost? It's not necessarily accessible to do these types of instrument modifications. Which kind of brings me to my research about sample expansion via hydrogel. Instead of making the laser smaller, we're going to make the tissue bigger, indicated. So the way we do this is we'll have our mouse brain fresh frozen, and we will section it, mount it to a slide, but instead of applying a matrix, we are going to polymerize a hydrogel on top of and throughout the tissue. And we're going to take advantage of the fact that the hydrogel, when placed in water, will isotropically expand in all directions. What we're going to do is covalently anchor analytes of interest and structural proteins to the hydrogel such that when the tissue and hydrogel are in water, they both expand. As you can see, certain structures, such as the corpus callosum here, is seen in the same configuration of our brain. Of course, that's a PowerPoint icon, or I think a bio-render image. But you can imagine then if we're supposed to do an IMS experiment with the same raster width on one tissue versus the other, we of course have many more pixels here, with the pixel density increasing by the square of the linear expansion factor. And this is what it looks like in real life, and kind of the features I was talking about. We can see white matter in the corpus, white matter in the cerebellum. We can see the corpus callosum, the striatum, the fimbriae. They're retaining their relative configuration. And of course, this is just visual, but we'll show some IMF data later on. And kind of as mentioned, it's inspired by expansion microscopy. So this kind of workflow of expanding tissues with hydrogel came about with fluorescence, first and foremost. So there's a good body of literature to draw on. And there have been efforts to move this to IMS, not only our lab, but several great publications and posters. This recent ASMS, I encourage people to look into as well as chat to me if you're interested in this. And hopefully, in showing how this is done, I can show how inexpensive and easy to implement this would be for any lab, really. So to go more in depth on how this is done, the first step is really a light fixation step, as well as preparing our tissue to be anchored to the hydrogel. And this is done using acryloyl X, which is a really simple molecule. It's an NHS ester attached to an acrylamide. The idea being, we can exploit the NHS ester chemistry and convert primary amines into acrylamide here. And if you're familiar with acrylamide, you probably know it's a very common monomer used in hydrogels, and that's our next step. We're going to pipette a hydrogel solution on top of our tissue that's going to consist of acrylamide and acrylate. And these are going to undergo free radical polymerization to form these long polymer strands. We're also going to have a cross-linker N-methylene bisacrylamide in there, which is going to join these polymers together to make a 3D polymer network. The key thing is we're not only going to incorporate the monomers we've added into these polymer strands, but also the acrylamide we've added to structural proteins, which effectively links our tissue into this 3D network. At that point, keep forgetting I have bubbles for each section. At that point, what we need to do is actually digest our tissue. As you can imagine, tissues are not homogeneous in any sense. They're, of course, very heterogeneous, have different regions with different densities of cells, structural proteins, etc. So if we were to expand as is, we get very uneven expansion and ultimately tissue tearing. So we need to mechanically homogenize the tissue. And what we can do is use proteases, as I mentioned, okay, trypsin, etc. But people can also use surfactants, heat, etc., chaotropic agents, depending on what you're looking at and really what is important for the structure of your tissue. And at that point, we can actually just put our sample in the water and expand. And one important point, just because I've gotten a lot of questions on this, is I am showing this on the slide here, but actually after the gel formation and cutting the excess gel, you can actually just grab the gel and pull it off the slide. So what you have is essentially like a rectangular block with the tissue on one side of the block. So I'm showing this on the slide. And of course, if you put in the digestion off the slide itself, your gel is really much separate from the slide during the expansion step, just sitting by itself with tissue on one side of the gel. And at that point, hopefully this looks familiar because this is our normal IMS workflow. We can take this gel, we can desiccate it onto an ITO slide, apply matrix, and then do multi-IMS. So we applied this to mouse brains looking at lipids, which kind of addresses one of our high spatial resolution concerns, right? Lipids ionize very easily in IMS, and their distributions are well known. And the first thing we wanted to see, of course, this is a long sample preparation. We wanted to address another concern, delocalization, of course, right? Most IMS sample prep is one day, it's several days, 2 or 3 days. We want to see our lipids moving around. So you have expanded brain here, 2x expansion. So not a lot of expansion right now. Hopefully we can visually see the increase in size from an unexpanded brain. This is the serial section. So we got one section, expand it, and then the next section immediately after, we leave unexpanded as a control to compare. And for any expansion data, I'm going to have the expansion factor up here, the raster width the image is taken at, and then the effective resolution of expanded tissue. So this is on sulfatide D42.2, and we can blow this up to get a look at it, just to put it at the same size, and just kind of basically looking at it, we can see our sulfatides are not massively delocalized to the tissue or different parts of the brain. They're very much still in the white matter. And we can continue to do this, of course, for different lipids, like the ceramide PEs, PCs, and I did it for 20, 30 lipids. There's 4 here, so we can look at them more closely, but negative mode, positive mode, etc. But this is still very vague, non-specific. So to get more quantifiable metrics, We can do the same thing. We took unexpanded tissue of serial section, expanded, and this is looking at the mouse brain cerebellum, and this looks at PC320. And again, we can use MATLAB and then register these two images of cerebellum on top of each other, allowing us to calculate PCC scores and residuals, again, across different structures and lipids. Generating here, we got a PCC of 0.89, but we got anywhere from high 0.7 to 0.9, depending on lipid and structure. We can also generate a residual score and image, which is the absolute intensity difference between the expanded and the unexpanded tissue. And the image is from, subtracting one from the other, which really highlights the differences we saw, which allows us to look at or investigate whether these differences are due to the fact that the serial sections not in fact the same section, sample handling, or artifacts from expansion, or just due to the fact that one is higher resolution. We don't expect them to look exactly the same. Another means, so we can see a lot of this difference is in the granular layer, where we can see a little more detail in the expanded tissue. And another way we can also investigate these results is with our serial section, we can wash off the matrix afterwards and apply an H&E stain, which provides an orthogonal means also at higher spatial resolution to investigate some of these differences. So we can see the granular layer stronger, or how this difference very much matches the shape of the granular layer we see here, as well as smaller features such as this fissure, which we weren't able to see as well in the unexpanded tissue. We can see there's a major difference in signal, as it was fairly visible in the expanded tissue. So that speaks to spatial distributions and how we maybe went about looking at differences. But we can also look at chemical fidelity in that lipids, of course, chemically are dominated by their fatty acyl tails, which really indicates their chemical properties. But they do all have distinct head groups, which vary quite a bit. Here's just smattering PIs, PAs, PCs, and PSs. So we wanted to see if during the expansion process, these lipids were maybe selectively either unrepresented or overrepresented during the expansion process. So we used IMS and identified essentially all the lipids we could in expanded versus unexpanded tissue. And this is just negative mode. We have similar data for positive mode. So this is around 150 lipids or so across a variety of classes. And we see remarkable parity. Again, this is just for a 2x expansion, so there's not so much signal dilution from just expansion, but throughout the gelation process and that 2x expansion, we still see not a major loss of identified lipids, and more importantly, the proportion of each lipid class maintains relatively the same. There is no one specific class that is overrepresented or completely demolished during the expansion process, which is corroborated when we look at a spectra synthesis. With spectra from like 700 to 850 and a positive mode range, which is where we're going to see a lot of these myelins, etc., A's. And for any major peak in the expanded, we do see a correspondingly unexpanded. Pretty great, but we do see populations of peaks that only exist in the unexpanded. If we go ahead and identify those, these are ethnopotassiated adducts, and not seeing them makes sense as the expansion takes place largely in water, so it's an unintentional washing step, washing away these potassiated adducts. And more importantly, these same lipids are identified still as protonated or sodiated adducts. You see blue to blue, yellow to yellow, etc. So given that we confirmed that, we then proceeded to use this to increase our spatial resolution. So on our left here is, again, mouse brain cerebellum. I've labeled the cell layer as white matter for Kinsey cell layer, granular layer, and molecular layer. I'm showing PC320 in blue and PC361 in green. And on the right is our unexpanded tissue. On the left is expanded. And let's blow this up to kind of give us a better sense. So this is a 4x expansion, which is the max we can do with this specific hydrogel composition, which brings us down to a 3.75 micron effective resolution. And visually, we can see even with the white matter, the amount of pixelated the borders look here, as well as I think probably the starkest is the Purkinje cell layer I mentioned again. So this coloring maybe isn't the best. We saw it previously with that other color scale, but you could tell it's kind of a dotted line, and we kind of see the same sense here, whereas here each individual Purkinje cell is a lot more clear. And like I mentioned, this is a 4x expansion, but we can modify the hydrogel composition to do a 7x expansion, potentially even more. We believe this delta is up to 10x. This was actually some work that's being done by an RAU student from France in our lab, Nana Kapi. She's doing some great work with these higher expansion factor gels, which brings us down from 15 micron, which is our limit, down to 2.1 effective resolution. So here's our unexpanded tissue with a larger portion, but I've kind of highlighted the region this is, looking at the same lipids. And again, this is something that's been big for our lab. Like I said, a lot of our IMS has been more tissue structure level in terms of what we analyze and look at. But there's only so much we can see. If we zoom in, blow this up, I think we can appreciate the same differences. Below, let's say, the granular layer, the amount of detail we're able to see is a lot greater in the expanded tissue, especially since the granular layer, familiar is filled with these granule cells, cell bodies, soma. We expect a lot of heterogeneity with each the amount of cell bodies there, which is not really, you can kind of tell in the unexpanded, but it's still kind of a blur. Whereas here we can see a lot more detail, which prompted us to do fluorescence microscopy in line with our expansion work. Staining the tissues or the gel tissues before expansion, as that is the original workflow for expansion using fluorescence microscopy. So this is just a DAPI stain, but again, comparing the same regions, you can kind of see the same general structure here. And if we go back and look at the Purkinje cell layer, so this is a combination of DAPI and cell mask of membrane stain here, and our IMS on the bottom, with the coloring a little different. Green is now PC320, and red is PC361, just because I think the contrast is easier to see. Not only can we see Purkinje cells, we can see the shape, the conflated kind of like pumpkin kind of, and we see that very much in our microscopy as well. And in fact, we can see one here, we can see this kind of longer Purkinje cell. We can line these up across. And again, just to throw back to what our Purkinje cells looked at, 15 micron versus our 2.1 micron now. So let's kind of sum up. Expansion is a sample-based methodology for increasing resolution that hopefully I showed was easy to implement. Also being sample-based, we presumably could stack it upon other instrumentation-based means of increasing resolution. Hopefully I've shown how it can be viable for lipid analysis, as well as ways to assess different structures, different analytes, different methods across mouse brain, and the expansion factor. We got 4x and 7x, and that gel we have expanded it up to 10x. We unfortunately just didn't have an image in time for this presentation, but up to 10x currently, and or 7x as shown at least. And in terms of future work, we definitely want to work more on our microscopy, getting more informative stains to get us more informative information, be able to identify structures at that smaller resolution, as well as increase our expansion factor further, and apply this to maybe some of our collaboration work, and see what we can see again at that higher spatial resolution. Of course, I'd like to acknowledge Dr. Prentis and the entire Prentis lab for their help with this project, collaboration, ideas, and of course, our funding. And yeah, be open to any questions.
Awesome. Thank you for sharing your incredible research with us. It's really impressive to see how you can take some of these approaches and then expand of small foci. So we'll open up to questions from the audience. Again, there's a button at the bottom of your screen that says Q&A. So just drop any questions there and we'll start working through them. So I'm going to give you the first one to get it going. And that is that one of the benefits of IMS, right, is that we can do this large, unbiased profiling of tissue. And you have the ability to show a lot of different analytes and their spatial distribution. And a lot of the times, right, if we're talking to someone who doesn't understand the field, we might call this like a multiplexed IHC. And then what the power that you've shown is that if you have a small foci, you can make it 7 to 10 times bigger, right? And then we can get that unbiased measurement of something very small. So my question for you is, how big do you think we could go? And at what point do you think that the benefits maybe outweigh potential artifacts or other things that might come into play in this tissue prep method?
Yeah, so there's a couple of key things. So in terms of like what you can actually physically do in terms of expanding the gel, I think you could get to 20x, but then kind of what you said about artifacts, like is that reasonable what we're looking at? Because like you can push it 20x from 5 micron, that's like 250 nanometers. But is that reasonable to say like what we're looking at is like actually things of that size, like organelles of that size? So one major hurdle I think we're going to have to see, there's two things depending on what you're looking at. So these lipids are physically retained in the gel, so they're not covalently cross-linked as other analytes, which as we expand further and further, I think there's a concern for whether they would maintain in the gel as kind of the pore size gets bigger and bigger. One solution people had is like iterative expansions. You expand the gel, and then you actually, it has a reversible cross-linker, and you can expand another gel on top, or another gel on top of that kind of. So I guess the pore size remain the same, expanded. So that's one way to deal with it. I think a more concerning one, something that I don't know if I have a complete answer to, is a lot of expansion, since it is such a intensive workflow compared to normal IMS. We actually section at 30 micron versus like a 10 micron, and microscopy like even lower, 5-7 micron for, in fact, like if we're looking at single cells or organelles, like, you know, you don't want multiple layers of those, right? So then if you're talking about 20x and organelles, like what does that look like with a 30 micron tissue? And can we maybe potentially decrease that size and, you know, perform
I'm going to
these
ask you, a tough follow-up. And it's about ionization efficiency. But as you get a thick hydrogel, do you see that you have any trouble getting ions into the gas phase, or do you have any like background from your proteinase K? Is there any like technical things that make it hard to get those ions in the gas phase?
Yeah, so this really comes down to doing lipidomics again, because lipidomics is really easy. So two parts. One, in terms of other reagents you might use, there is like extensive washing, so there shouldn't be any proteinase K in there, hopefully not. But again, the lipids very easy to see, and we're using Dan lipids, but there's even other matrices we want to use that are even more efficient than Dan. So like being able to use a small laser spot size, so that's no problem. But we have seen with other animals that we're working on, like using higher laser power, you do start to see like polymer or adduct peaks that do become an issue. So that might also be another issue, especially if you want to talk about the untargeted power of IMS. Like look at all these analytes, you know, it's very, kind of like I said, it's very much dependent on the ionization, and then if you're going to get artifacts alongside it.
Yeah, so we actually have a question following up on that about your beam size. So with the expansion, can you do a Z-scan of the beam size that is smaller than the feature size in the Z-axis? That's a tricky question.
Yeah. Wait, so was a Z-scan of beam size smaller than? I'm not actually familiar with
the expansion.
Z-scan.
Can you do usually Z-scans up and down, right?
Yeah, that's what I was thinking. That's not really, because I know in microscopy, I guess you can get like.
But you're on a Solarix, right? So,
yeah.
So this is maybe a timsTOF-ish question, right? They do that. Anyway,
They do. Oh, okay, yeah.
Maybe we'll answer you offline. Great
Yeah,
question.
I would definitely like to follow up on that. That's a little bit on the but yeah.
Absolutely. All righty. So then the last question that we have here is, have you tested this technique for metabolites?
We haven't, but actually one of the publications I mentioned, TEMI, they actually really know, but they're really good for a lot of metabolites. So I would definitely recommend. Let me see if I can get my screen up again. I mean, all it has is the author's last name, but that should be more than enough to find the paper. Let's see. So they have done it for metabolites, which was honestly surprising, because at first I would have thought it's not possible for metabolites. Just given, again, you either need a covalently link or have physical retention in metabolites, of course, can be really small and really polar. This one, I mean, 2025. So I recommend looking into that for metabolite work. And they do kind of that, what I said, that iterative expansion, which, and they also do a very gentle digest, which might go into their ability to see metabolites as well. Yeah. So, yeah.
Are any of these works protein focused? Do you know?
Intact protein, no, but two of those actually. I can bring it back up.
Sorry.
No, that's okay. At least I'm on the slide right now. So Gamzee did proteins, but with labeling. So they used kind of Ambrogen label. And they were able to get that. Temi also did like a bottom-up proteomics, like spraying trypsin. Yeah. So definitely for like analytes availability, Temi will be, I think, the paper to look at, especially since it's so recent. I think it's kind of, I mean, the whole field is recent, but this is kind of like one step up. But it is also Banton done a little differently. They take a whole section and then
Yeah,
like a big chunk, and then they section that and that's all not it. So it's a little different than what I presented, but definitely worth reading if you're interested in this work.
absolutely.
Yeah.
Well, so I think that we'll transition in the last 5 minutes to just kind of the fireside chat portion where anybody can ask anything. And the seed question that we have for this for either Jacob or Christiana is that you've shown complicated workflows for spatial omics, and they're both amazing, right? And we know that it kind of takes a village when you get deeply into spatial biology. So did you find that you needed to reach out and include like new expertise for your team to accomplish some of the questions that you were asking in your research? So I'm thinking like pathologists, microscopy experts, like who did you reach out to help you come up with these solutions?
I can go. Yeah, kind of like what you said. One thing was polymers. There's like great polymer labs here at UF. Some learning people who work with hydrogel, so picking their brain for like how these work, things we can maybe do to characterize the gel. They have a whole polymer characterization lab. And microscopy is another big thing I mentioned so many times that IMS when we get to the small subcellular range, it's just not what we typically look at. We're typically looking at tissue structure level. So one, learning how to use our microscope effectively, like validate our various stains and antibody suggestions for different stains. Like I know something that's been big is single cell, like I mentioned, identification, and how we can use microscopy along with IMS to identify. So both of those have been really big for.
Yeah, so in our case, I'm lucky that I'm at the institute that has broad ranges of expertise, so from cancer biology to epigenetics to sequencing and so on. So we had local partners that would help out, very local partners. But I think one of the challenges was that, of course, we focused also on the protein that we had the tools to study also the best. So depending now on the pathways that you find, we also have targets related to metabolism, which are not necessarily now our comfort zone. So then, of course, this would kick out something completely new, which is on the list as well. Yes.
Do you have any suggestions for thinking outside of the box to answer your spatial biology questions for the scientists who might be here regarding designing experiments or finding new technology to acquire data? How did you get to this? How did you find these solutions to implement in your spatial systems?
Now you mean for example, in my case, how I discovered optoproteomics? Well, in my case, it was a bit of a coincidence through former conference contact, actually, that I was connected with on LinkedIn, and he was a former scientist, and he is now also the CEO of the company that has developed the optoproteomics technology. And this was in the time when I have found these bodies, and I had no idea how to study them spatially. And in parallel, he was looking for people on LinkedIn. And I saw the technology and I thought, okay, this is a perfect match. And then we reactivated the connection. So it was in a way coincidence, but also caused a little bit by going to conferences and talking to people.
Yeah, kind of similar. I mean, I know this is something actually Dr. French has worked a little bit on in grad school, but definitely for new ideas and how to better talking to collaborators, but also just reading papers, going to presentations. So I find if I'm just doing my same thing, it's very focused in, but getting new ideas. And then it's always frustrating when you find a solution. It's like, I'm in the literature the whole time. I just, didn't read the right paper. So I think that's done. Yeah. Reading outside of my comfort zone. Yeah.
Yeah, absolutely. And the power of this type of thing, right, where we all can come together and share our new technologies. So we are sadly drawing to the end of our hour together. So I want to thank both Jacob and Christiana for sharing these approaches that maybe you can try in your research. Of course, we know spatial omics has so many challenges in measuring small foci, and then both of these speakers highlighted an approach to analyze them within biological systems. So thank you again for both of you and to the audience for joining us today in our continuing webinar series on spatial proteomics and imaging. And we look forward to seeing you in our next session. So thank you all.
Thank you.
Thank you. Yeah.